comes to the fields of myelin imaging, you have MRI-based, and then you also have molecular imaging-based approaches using PET. And so looking at just generally comparing quantitative imaging of myelin using MRI, it's a very indirect measure, primarily associated with isolating the signal or magnetization exchange that occurs between the water that is trapped within the myelin wrappings and then the myelin itself as what is assumed the primary macromolecule substituent of the CNS. So it's a very indirect measure. And also importantly, you can't compare measures from different techniques. You can't take myelin water fraction imaging compared to MRI, to QSM, to magnetization transfer ratio, MTSAT, and homogeneous magnetization transfer, diffusion metrics. They're all specifically unique to their own bubble. Whereas when you look at the molecular imaging of myelin, you're introducing a radio tracer that associates with myelin itself, and then you can get some more direct measurements. Now, there are a bunch of different pros and cons between the two techniques, but what I won't go through them all, but what I will say in general is that where one technique has a limitation, it is usually the strength of the other technique, which provides a great opportunity to create some redundancy. If I were to be a pharmaceutical company who had invested a great deal of both time and monetary investment into a certain compound, I would ideally like to include both MRI-based imaging of myelin and PET-based imaging of myelin, specifically because not just, well, they're both looking at myelin, but conceptually they are approaching quantifying myelin from entirely different directions. And if myelination is observed, it just adds so much further credence to this observation if it can be captured in both techniques, because they're not approaching it from the same way. One's direct, one's indirect, based off entirely different assumptions. So there is great value in how they work together. Now, looking specifically at the molecular imaging of myelin, really started with Bruno Stankoff looking at carbon-11 BMB. There's been a few diaminostilbene derivatives put forth in animal models, mouse models, Cooper's-own mouse models, lysolecithin treated rat models, also looking at EAE in non-human primates, some spinal cord injury in rodents as well. But that also brings the idea of the difficulty, because much of the pipelining and development for these radio tracers, it really parallels that of drug development. So there are much more higher hurdles, because it is an injectable, to overcome relative to myelin imaging. But importantly, if you have a drug that interacts with a certain way, but fails to have a desired effect, but you know that there is an interaction, you can repurpose, which interestingly has been done in molecular imaging of myelin in the field of MS, where repurposed tracers that are more typically known in Alzheimer's disease to target amyloid beta aggregation and the A beta plaques. And this is really thought to be because the A beta plaques, you have your beta sheet stacking, which is a highly lipophilic environment. And so for a tracer to be able to embed in the highly lipophilic A beta aggregations, there's also this non-specific binding that occurs in the white matter. And that's believed to be off-target binding, at least from the perspective of AD, off-target binding that interacts with myelin. And there is myelin basic protein also has beta sheets. And so there's an interaction that's believed to be there. And so and since we've had BMB, there's been a lot of diaminostilbene derivatives, C-MIDAS being one of them, but then also CIC, Pittsburgh compound B. But then now there's been some second generation fluorinated tracers, and fluorinated tracers are very important because they provide applicability to PET imaging centers that don't have a radio pharmacy, a radiochem lab, cyclotron, which is necessary for carbon-11 labeled radio tracers because of its short half-life. So these fluorinated tracers can be created off-site, shipped to your lab, and then you can use them for imaging there. And there has been promising studies across most of these second generation fluorinated radio tracers, specifically the fluorinated versions of florbetapir and florbetapen have been the most promising, but I believe that there still needs to be validation, particularly in the repeatability and making sure that the methods are very comparable and how they're applied, to really lend credence to really picking out normalization area. One of the more difficult aspects of looking at the molecular imaging of myelin is if you want to do metabolite analysis, you'll need to have an arterial line placed, which is a rather invasive procedure that most patients are not entirely comfortable with. It requires an anesthesiologist, and it just kind of increases the threshold of clinical organization necessary. I mean, and it's also certainly a hassle for the patients. It's not comfortable. I've never had one done. There's a reason for that. I would not like to have one done. But also for the caregivers as well, for those with more advanced disease, their caregiver, it's also difficulty for the caregivers as well. So if it can be avoided, I think it should be avoided. And I think that we should approach technological considerations such that it can be avoided. And that should be done in the future as well. And so using florbetapir and florbetapen, I think next steps, characterizing treatment, seeing if there's potential for remyelination. I also think that there's some good in vivo dynamics of myelin patterns that are good proof of concept checks that should be done in these myelin imaging sequence. It's been shown across a few MRI-based myelin imaging techniques that there is a periventricular gradient of demyelination. Arguably, this was initially thought to be a partial voluming effects in the region, but it exceeds well beyond what would be the typical area of partial voluming effects in the parenchyma. And so this has been shown using MRI with MTR, notably myelin water fraction imaging, the PSI-MRI technique, and more. But also importantly, it's been shown using Pittsburgh Compound B from Verna Stankoff and Benedetta Bodini's group as well. So it's a good proof of concept that if you're applying a tracer, it should be able to capture this. And so looking at treatment effects and moving forward with applications for these fluorinated tracers as well. And also aiming to characterize the specific interaction between these tracers and myelin, again, believed to be the beta sheets and myelin basic protein, previously shown for PID. But it's important that we do characterize that. Just the more we know about these, the better. So there's promising horizons on the field of molecular imaging of myelin, particularly in MS. But again, think about that MRI-based and molecular imaging-based imaging of myelin. They're not at odds with one another, but they're important redundancies that if you have a capable clinical facility, you should try to do so. But certainly there is radiation involved with these with PET imaging, so that certainly limits its application. It is expensive, so using MRI as sort of a guiding tool for molecular imaging of myelin is important. Not only can it be used to potentially identify patients who would most benefit from acquiring PET scan looking at myelin, but also for enhancing the contrast. Bruno Stankoff and colleagues have just published a very interesting study using a combined PET MR, which again is a tool that not all clinical facilities have, but you can use MRI to enhance the resolution of PET, which is one of the limitations of PET imaging. You have a certain resolution defined based on your type of sensor, its size, just based on the annihilation of the radio probe itself. And so enhancing the resolution, but then also identifying regions that could specifically value PET imaging of myelin, mainly because there's a different dynamic of myelin that is and can be captured using PET. Again, I said it's approaching from an entirely different perspective, and identifying this heterogeneity of myelination in lesions, but across the parenchyma is very difficult. And one last point I would like to comment on is looking in the gray matter, and particularly the cortex. The cortical ribbon is really a wonderful word in that it really does characterize this very thin structure that is highly convoluted and very difficult to image. And so looking at the gray matter myelin content with both quantitative MRI measures and molecular imaging is highly difficult. And so focus on this area as you develop your tools, as you move forward clinically. Claudio Lucanetti, 2011, very wonderful histopathological study looking at cortical myelination, characterizing the lesion types. This was followed up by many groups. One in particular, my previous colleague, Katarina Minero, using 7 Tesla to characterize cortical lesions. So we know that there's a lot going on in the cortex, but there is much more to be discovered, and looking at not only Katerina and colleagues, Elena Harantha, looking at microglial activation in the cortex, but I would also like to see other types of tracers be applied within the cortex to really determine the role of the cortex and better characterize our understanding there. So yes, I think that just about covers a few things to consider with respect to the field of myelin imaging itself.